Welcome to the PubMed Daily Review for May 15th, 2026. Today we scanned 8,109 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5. Varied research methods and applications. Diverse studies showcase the development, application, and enhancement of varied research methods across multiple scientific fields. This cluster covers 1,304 papers. Representative work includes sperm whales habituate to research vessels engaged in photo identification. Effort produces after effects costly for others, but valued for self, and enhancing IRMAL-DC MSI spatial resolution through beam constriction with a ring-actuated iris. Trend 2 of 5. Cognitive function and mental health. This trend explores the interplay, mechanisms, and interventions for cognitive function and mental health across diverse populations. This cluster covers 1,293 papers. Representative work includes effectiveness of multisensory stimulation on cognitive function in older adults with mild cognitive impairment. Protocol for systematic review, methodological framework for the design and implementation of a U.S. Latino-Hispanic digital brain health program. User-centered design approach, and subregional activity in the dentate gyrus is amplified during elevated cognitive demands. Trend 3 of 5. Clinical interventions and cancer therapies. These papers collectively advance cancer care through novel clinical interventions, therapeutic strategies, and improved delivery and management approaches. This cluster covers 1,019 papers. Representative work includes a natural language processing framework for structuring and visualizing clinical trial eligibility criteria at scale. Protocol for a quantitative study. Disentangling positive effect heterogeneity in a digital physical activity intervention for young adult cancer survivors, a group-based multi-trajectory modeling approach, and a Phase 1 oral mass balance and combined intravenous 14 C microtracer study to characterize the absorption, metabolism, excretion, and pharmacokinetics of anti-tumor drug E7386 in humans. Trend 4 of 5. Molecular biology and pathogen studies. This trend investigates molecular mechanisms, pathogen biology, and host interactions using diverse genomic and experimental models. This cluster covers 1,761 papers. Representative work includes identification of the regulatory elements and protein substrates of lysine acetoacetylation, genomic signatures of dominant clones, and evolutionary divergence of Acinetobacter baumannii in Thailand, and establishment of human coronavirus HKU1 infection evaluation system using apical-out airway organoids. Trend 5 of 5. Health services and patient care. This trend explores optimizing patient care and health service delivery through clinical insights, technology, and public health strategies. This cluster covers 2,732 papers. Representative work includes cost-effectiveness of Internet of Things-based management of home non-invasive positive pressure ventilation in patients with chronic obstructive pulmonary disease and hypercapnic chronic respiratory failure. Trial-based economic evaluation, vaccination-related applications, and healthcare professionals observe changes in human papillomavirus vaccine hesitancy. Cross-sectional survey and determinants of willingness to receive health information from neighborhood food and beauty establishments. Cross-sectional study. Up next, our first paper out of 5, a NeuroD1 AAV-based gene therapy for functional brain repair in Alzheimer's disease-like non-human primate model by Zhouquan Jiang and Wenliang Lei from State Key Laboratory of Bioactive Molecules and Drugability Assessment, Guangdong Basic Research Center of Excellence for Natural Bioactive Molecules and Discovery of Innovative Drugs. Guangdong Key Laboratory of Non-Human Primate Research, State Key Laboratory of CNS Regeneration, Ministry of Education, GHM, Institute of CNS Regeneration, Jinan University, Guangzhou, Guangdong, China. Let's break down its key contributions. Alzheimer's disease remains a devastating condition with a critical need for new treatments, especially those that can regenerate brain tissue. A novel approach involving NeuroD1 gene therapy has shown promise, but its efficacy in complex animal models, particularly non-human primates that closely resemble human disease, was unknown. This research aimed to fill that gap by evaluating this gene therapy in a sophisticated NHP model of Alzheimer's. Our study utilized a sophisticated non-human primate model of Alzheimer's disease, specifically engineered with hippocampal H-tau overexpression to closely mimic human pathology. We employed a comprehensive suite of techniques, including advanced imaging like MRI and PET, alongside CSF biomarker analysis, behavioral tests, and RNA sequencing. The primary aim was to assess the therapeutic potential of NeuroD1 AAV-based gene therapy, exploring its impact on neuroregeneration, synaptic function, and inflammation. The results were highly encouraging. We observed that the NeuroD1 gene therapy effectively prevented neuronal damage and degeneration, a critical finding. Furthermore, it significantly enhanced spatial working memory, indicating improved cognitive function. The therapy also reduced neuroinflammation, inhibited hippocampal atrophy, and restored CSF-AD biomarker levels, all pointing towards a comprehensive therapeutic effect. These findings collectively demonstrate a remarkable repair and restoration of brain structure and function. This study represents a significant step forward, demonstrating that NeuroD1 AAV-based gene therapy holds strong potential as a neuroregenerative treatment for Alzheimer's disease. The use of a non-human primate model lends considerable weight to these preclinical findings, suggesting high relevance to human pathology. While incredibly promising, the next crucial step will be to translate these results into human clinical trials to assess safety and efficacy in patients. Up next, our second paper out of 5, HEX, a MASP for mapping the whole tissue spatial proteome and the intra-brain distribution of monoclonal antibodies by Shirhan Hua and Jun Chu. Let's break down its key contributions. Spatial proteomics, which maps proteins across tissues, is crucial for understanding biological processes. However, current methods struggle with resolution, practicality, and throughput, limiting our ability to fully explore complex tissues like the brain. This paper introduces a groundbreaking new technique to overcome these challenges. Our team developed HEX-MASP, a next-generation spatial proteomics technique. It features innovative hexagonal microwells created with optimized 3D printing, ensuring high resolution and robust sampling. This design significantly enhances sample preparation and analysis, making it more practical and effective for whole tissue studies. Applying HEX-MASP to mouse brains, we successfully mapped over 6,000 proteins across the entire tissue with unprecedented spatial accuracy and excellent data quality. This allowed us to uncover critical regional details previously missed and identify many new regionally enriched proteins. We even mapped the distribution of antibody therapeutics for the first time. HEX-MASP represents a robust and scalable platform, poised to revolutionize whole tissue spatial proteomics. It not only enables the discovery of novel protein distributions and drug mechanisms, but also holds immense potential for advancing neuroscience and drug development. We believe this technique will open new avenues for research and therapeutic insights. Up next, our third paper out of 5, AI-powered programmable virtual humans toward human physiologically-based drug discovery by Yu Wu and Lei Xie from School of Pharmacy and Pharmaceutical Sciences and Center for Drug Discovery, Northeastern University, Boston, Massachusetts, USA. Let's break down its key contributions. Drug discovery is a complex and often inefficient process. While AI has shown promise, current methods often fall short, struggling to accurately predict how new compounds will perform in humans. Existing digital twin technologies are also limited to later stages of development, leaving a crucial void in early-stage research. This paper explores how we can bridge this gap. This paper proposes a novel approach, AI-powered programmable virtual humans. These aren't just simulations, they're dynamic, multi-scale models built upon advances in AI, high-throughput assays, and detailed single-cell and spatial omics data. The core idea is to create virtual environments where novel compounds can be tested directly within a human physiological context, something impossible in the real world. While this is a conceptual paper, the proposed virtual human approach promises significant advancements. It aims to dramatically improve the prediction of clinical outcomes, bridging the translational gap that often hinders early-stage drug discovery. This means we could optimize drug efficacy and safety much earlier, enabling virtual experiments that are simply not feasible in traditional lab settings. This paper presents a compelling vision for the future of drug discovery, moving beyond current limitations with AI-powered virtual humans. While this is a conceptual framework, its successful implementation would require substantial advancements and integration across various scientific and technological domains. Ultimately, This approach has the potential to fundamentally transform how we discover and develop new medicines, making the process faster, more efficient, and more predictive. Up next, our fourth paper out of 5. Emergence of vaccine-derived poliovirus strains from the novel oral polio vaccine in the Central African Republic by Joel W. Dote and Mail Basad from Institut Pasteur de Bangui, Laboratoire de Virus Enteriques / Rougeole, Bangui, Central African Republic. Let's break down its key contributions. Poliomyelitis remains a global health threat, especially in vulnerable regions like the Central African Republic. Historically, the live-attenuated oral polio vaccine, while effective, sometimes led to the emergence of vaccine-derived polioviruses, causing new outbreaks. To address this, a novel oral polio vaccine, NOPV2, was developed with enhanced genetic stability. This study investigates how NOPV2 performs in a real-world setting, specifically looking at whether it truly prevents the emergence of these problematic vaccine-derived strains. Researchers conducted routine poliovirus surveillance in the Central African Republic from late 2021 to late 2023. They collected stool samples from patients and contacts, as well as wastewater, to identify circulating poliovirus strains. Using advanced genetic techniques like Sanger and whole genome sequencing, they meticulously characterized these isolates. The goal was to understand the genetic evolution of both historical and novel vaccine-derived polioviruses, particularly focusing on how NOPV2's engineered stability held up against real-world viral dynamics. The surveillance identified 54 vaccine-derived poliovirus type 2 isolates, originating from 8 distinct emergence events. Crucially, 4 of these emergences were derived from the novel NOPV2. While NOPV2 showed stability against genetic drift in its primary attenuation site, a significant finding was that some NOPV2 derivatives had lost their attenuation through recombination with other enteroviruses. This included the detection of double recombinant NOPV2-derived isolates, which had completely lost their attenuation determinants, marking the first reported instances of such reversion from NOPV2. This study provides crucial insights into the real-world performance of NOPV2. While it confirms the vaccine's improved genetic stability against direct reversion, it also reveals a significant challenge. Recombination with other enteroviruses can still lead to the emergence of dangerous vaccine-derived polioviruses. This means that achieving polio eradication requires not just monitoring poliovirus, but also a broader surveillance of related enteroviruses, particularly in high-transmission areas. Future vaccination strategies may need to consider these recombination risks to ensure lasting success in polio eradication. Up next, our 5th paper out of 5, Tropism and Replication Competence of Cattle Influenza H5N1. Genotype B3.13 virus in human bronchus and lung tissue by Kenry P.Y. Hui and Michael C.W. Chan. Let's break down its key contributions. Hello, everyone. Today, we're diving into a critical public health topic: the emergence of H5N1 influenza viruses from cattle. These viruses have recently caused mild infections in humans, raising concerns about their potential to adapt further. This paper addresses a crucial question: how do these cattle-origin H5N1 viruses behave in human respiratory tissues? To answer this, researchers used an ex vivo model, studying human bronchus and lung tissue explants. They employed a range of techniques, including viral replication assays, immunohistochemistry, and gene expression analysis, to thoroughly characterize the viruses. The goal was to understand the virus's tropism, replication, receptor binding, and the immune responses they trigger in human cells. The results are quite telling. In human bronchial tissues, these cattle H5N1 viruses showed moderate replication, notably higher than avian H5N1 strains. In lung tissues, their replication was similar to the common H1N1 PDM virus. Crucially, they acquired a partial affinity for human-like alpha-2,6 linked sialic acid receptors, indicating a step towards human adaptation. They also induced robust pro-inflammatory responses, falling between seasonal flu and highly pathogenic avian strains. So what does this mean for us? These findings suggest that cattle-origin H5N1 viruses are showing signs of increased adaptation to humans, posing a potential health risk and raising concerns about human-to-human transmission. While these ex vivo studies are powerful, we must remember they're models. Therefore, Continued research and vigilant surveillance are absolutely critical to track these evolving viruses and prevent future outbreaks. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.